Hi everybody, I'm glad you're with us. Today we're going to be looking at the story of RB and E2F, and it's going to be terribly exciting. But before I do that, I thought we'd just go ahead and navigate through this Presley just a little bit. So let's not forget that this is divided into the different parts of the cell cycle. You have G1 here, S synthesis there, G2 or gap 2 there, mitosis, which is not cellular division, cytokinesis, which is the separation of two cells, and you actually have a couple extras. You have G0 out here, and you have apoptosis over here. You also have a mitogen or signal coming in here. Let's click randomly on a few things. This is the story of a checkpoint involving some molecule called P53. I wonder if it is important for cancer. This is also a story. It involves a receptor, RAS, MAP kinase, and intermediate gene expression. I wonder if any of this is informational, important stuff. I wonder what mic production has to do. Well, excessive mic production has to do with anything. It's got that protein P53 again in it, in cell cycle and apoptosis. Interesting. Alright, now on to our story. So, our story is pretty simple. There's a protein E2F, and it is going to bind to DNA. Well, let's look at another diagram here. So E2F here binds to the E2F box, which is a transcription factor for several genes. So E2F binding to DNA causes the transcription and then translation of several genes. When E2F is free, it can bind to DNA. However, it's often bound by a protein called RB, named for the disease it was found in, and that's retinoblastoma. So when RB is phosphorylated, when several phosphates are added to it actually, by active G1 CDKs, then RB becomes inactive and E2F is free. So let's go over that one more time. RB phosphorylated, it can no longer bind to E2F. E2F is free, and E2F is going to go ahead and bind to DNA, as we see right up here, and it's going to cause transcription and translation of several genes, specifically ORC genes, CDC6, MCM, and a bunch of others. That's the easy part. Here's where it gets a little bit confusing. E2F is going to cause the transcription and translation of S-phase genes, specifically these. E2F is also going to cause more expression of itself. G1 and S cyclins also phosphorylate RB, and S cyclins and CDKs activate DNA synthesis, but also activate G1S CDKs, which go back and phosphorylate RB. The result is three positive feedback loops. This one here from E2F, which causes the production of more E2F, G1S cyclins, which phosphorylate RB, and active S CDKs, which phosphorylate G1S CDKs, which then phosphorylate RBs. So that's the basic story. Important parts are inactivated RB means activated E2F. E2F is a transcription factor, and there are three positive feedback loops here. That's it.